EO2463 is what we call an off-the-shelf peptide vaccine. The vaccine is made up of 4 peptides, which are pieces of proteins that are derived from bacteria that we all have in our gut microbiome. These peptides, they have molecular mimicry, so they resemble antigens that are present in B cells, specifically CD20, CD22, CD37, and the BAFF receptor. The key innovation of this concept is to leverage pre-existing cytotoxic memory T cells that we have previously developed and target these against the B cell lymphoma tumor. SIDNEY was a phase 1/2 trial enrolling patients who were HLA-A2 positive with relapsed and refractory follicular or marginal zone lymphoma who had received at least 1 prior line of therapy. So the peptide vaccine is combined in this study with 2 other agents, rituximab and Revlimid or lenalidomide. So as of May 5th, 2025, 22 patients were evaluable for safety, and their baseline characteristics showed essentially that most patients had a median age of 60, performance status of 0-1 in 82 or 18% respectively. Median time from primary diagnosis was 6 years. Most patients had 4 sites of disease, with ranging from 1 to 16, and 17 patients had no POD24. In regards to the safety profile of this compound, we found overall that the combination of the peptide vaccine with R² was well tolerated. The most common adverse event was a local administration site reaction, which occurred in approximately 68% of patients. Hematological toxicities were as expected for patients being treated with R², including neutropenia in 64% of patients and thrombocytopenia in 32% of patients. Severe grades 3 and 4 adverse events were primarily hematological, with neutropenia being the most common at 32%. It's important to mention that only 1 patient discontinued due to an adverse event. As of the time of this presentation, 6 patients had discontinued the therapy for progression of disease, while 32% completed all planned therapy and 27% were still on treatment. In terms of the efficacy of the compound in combination with R², we found out of the 20 evaluable patients when combining cohorts 1 and 4, 3 out of 9 patients in cohort 1 showed a response to single agent EO2463 at week 6, which is the end of the monotherapy treatment phase. And this is prior to the addition of lenalidomide. Adding the lenalidomide increased the response rate substantially. By week 19, which is prior to the addition of rituximab, we observed a 60% overall response rate, half of which were complete responses. 5 patients in complete response continued without receiving rituximab, which was optional in that case. The overall response rate at week 42, which is after the addition of rituximab, increased to 65%, including 60% of patients in complete response. The median time to complete remission was 23 weeks, and we saw responses across all high-risk disease groups, including patients who were positive for Gauss criteria, high FLIPI score, and POD24. When we evaluated the immunogenicity of the compound, we noticed a significant association between the generation of specific immunological responses and the likelihood of achieving a complete response, which speaks to the mechanistic capacity of these peptide vaccines to generate lymphoma-specific immunity. In conclusion, the EO2463 compound in combination with lenalidomide and rituximab is well tolerated in patients with follicular and marginal zone lymphoma. The EO2463 generated a fast, robust, and durable expansion of EO2463 mimic peptide and target B cell specific CD8 positive T cells. And the EO2463 monotherapy showed direct anti-lymphoma activity within as short a time frame as 6 weeks, which is only after 3 administrations of the peptide vaccine. In our preliminary data, EO2463 seems to support a higher than expected complete response rate when compared to patients historically treated with rituximab and lenalidomide alone. We also found that EO2463-driven expansion of specific cytotoxic T cells correlates strongly with the likelihood of achieving a complete remission for patients treated in this trial. We also propose the optimization of the ER squared schedule by moving the rituximab to an earlier stage, and feasibility testing with rituximab starting as early as week 7 is already underway. Finally, we found that immune response might be developed as a potential biomarker for patients treated with EO2463, and these results support further development of EO2463 in patients with lymphoma.